So our primary discussions in this video is going to be glycogen. So we said before that glucose is the most important monosaccharide, thus it's important that our cells have the ability to store glucose. The way that we store glucose is by converting it into glycogen. So glycogen is found in both the liver and the muscle, and it's used as a rapid way to get glucose when the cells need it. So muscle is going to break down glycogen to glucose to use it in the process of glycolysis to generate ATP, and liver can also break down glycogen to form glucose that it could either release it into the cells, into the blood to reach other cells, or or the liver cells can use it. Glycogen is just long chains of repeating units of glucose. So if we take a deeper look at the structure of glycogen, we can see that glycogen is made up of monomers of glucose. So these glucose molecules can be connected with each other by two different bonds. The first one is called 1,4 glycosidic bonds, and the second is called 1,6 glycosidic bonds. So the 1,4 glycosidic bonds are just going to create straight chains of glycogen. The 1,6 glycosidic bonds are going to be responsible for the formation of the branches on the glycogen molecules. And something really important to know is that when we look at the glucose molecule, it's important to know how we number them, so we know what type of bond it is. So we're going to start with the first carbon right here, and then we're going to make the way all the way to the sixth carbon right here. So the 1,4 glycosidic bonds is going to be between carbon number 1 and carbon number 4 on two different glucose molecules, and the 1,6 glycosidic bonds are going to be between carbon number 6 and carbon number 1 between two glucose molecules. So glycogen synthesis comes out of the glycolysis pathway. So right here we have the glycolysis pathway. So what's going to happen is that the glucose 6-phosphate that's part of the glycolysis pathway is going to come out and be converted into glycogen. So now we're going to talk about the specific steps that are needed in order to synthesize glycogen. So glycogen synthesis is a 4-step process. We're going to start with glucose 6-phosphate, and we're going to end with branched glycogen, like we can see right here. So the first step is going to be the conversion of glucose 6-phosphate to glucose 1-phosphate. Remember that glucose 6-phosphate is going to come from the glycolysis pathway. So what happens here is that we just move the phosphate on the glucose molecule from the 6th carbon all the way to the first carbon. The next step in glycogen synthesis is going to be the conversion of the glucose 1-phosphate into something called UDP glucose. And this is going to be catalyzed by the enzyme UDP glucose pyrophosphorylase. So what happens here is that we're just going to add a UDP molecule onto the glucose 1-phosphate. So this right here is going to be the UDP molecule, and this is going to be the glucose 1-phosphate, and we just put them together. So the UDP that is added is going to be the same UDP that is used in RNA synthesis. So the next step would be the conversion of UDP glucose into unbranched glycogen. And this is going to be catalyzed by the enzyme glycogen synthase. Now this is a very important enzyme in glycogen synthesis. So what happens is that we take basically a few molecules of UDP glucose, and we're going to cut off the UDP, and we're going to tie in the two glucose molecules together to get unbranched glycogen. Now the unbranched glycogen are all going to be formed by 1,4 glycosidic bonds. So the fourth and the final step in the formation of glycogen is going to be the conversion of unbranched glycogen into branched glycogen, and this is catalyzed by the enzyme called the branching enzyme. So as you can see in the picture, we converted the unbranched glycogen right here, and we added a branch of glycogen at this point, and this is going to be formed of a 1,6 glycosidic bond. So after we've discussed glycogen synthesis, now we're going to talk about glycogen breakdown. And this is the mechanism by which our cells break down glycogen to get glucose, and how our cells use glucose to make energy, to make ATP. So there are two important enzymes that are used in glycogen breakdown. The first is called the debranching enzyme, and the second is called the glycogen phosphorylase. So the debranching enzyme is going to work on the alpha-1,6 glycosidic bonds, and it's going to convert branched glycogen into unbranched glycogen. The glycogen phosphorylase enzyme is going to work on the alpha-1,4 glycosidic bonds, and it's going to convert them all the way up to glucose 1-phosphate. Now once the cell develops glucose 1-phosphate, it's going to convert it back into glucose 6-phosphate. Now glucose 6-phosphate has the choice of either entering glycolysis to make ATP, or it could either be converted back into glucose in the process of gluconeogenesis, and this is catalyzed by the enzyme glucose 6-phosphatase. So we said before that there are two tissues in the body that have glycogen. The first is going to be the muscle, and the second is going to be the liver. So once the muscle develops glucose 6-phosphate, muscles could only use glucose 6-phosphate for glycolysis and to make ATP. The muscles cannot undergo gluconeogenesis. In the liver, the cells have two choices. It could either use it for glycolysis to make ATP, or it could synthesize glucose, depending on the body needs. So now we're going to talk in a little bit more detail about the enzymes that are used in glycogen breakdown. The first enzyme that we're going to talk about is glycogen phosphorylase. Now, like we said before, glycogen phosphorylase is going to work on the unbranched glycogen chains, and it's going to work on the alpha 1,4 glycosidic bonds. So it's going to work on these bonds right here, and it's going to break the glycogen to yield basically free molecules of glycogen of glucose 1-phosphate. So something important to know is that the enzyme glycogen phosphorylase is actually stabilized by vitamin B6. So the glucose 1-phosphate that is formed, like we said before, could either be used in glycolysis or gluconeogenesis. So the process of glycogen breakdown is going to start with the enzyme glycogen phosphorylase. It's going to work on the alpha 1,4 glycosidic bonds, and it's going to break these bonds to yield free glucose 1-phosphate that we can see right here. Now, it's going to keep doing this until we reach about 3 glucose molecules that are connected with an alpha 1,6 glycosidic bond. Now, after this point, glycogen phosphorylase cannot function, and the remains, which are these 3 glucose molecules, are called limit dextrans. So at this point, we're going to need another enzyme to start functioning, and that enzyme is going to be called the debranching enzyme. So the debranching enzyme is going to work on these limit dextrans, and the way that it works is that it's going to remove 2 glucose molecules, and it's going to add it to another limit dextran, and it's going to release the glucose molecule that is attached by a alpha 1,6 glycosidic bond, as we can see right here. So it's going to add 2 glucose molecules to this limit dextran, and then it's going to add another 2 glucose molecules to this limit dextran. Now, after we elongate this limit dextran, the glycogen phosphorylase enzyme could come along and break the alpha 1,4 glycosidic bonds to yield more glucose 1-phosphate. Okay, so now we're going to talk about the hormonal regulation of glycogen synthesis and breakdown. So we're going to discuss how hormones affect either the process of glycogen synthesis or the process that promotes glycogen breakdown. So there are 3 important hormones that we're going to talk about. It's going to be glucagon, epinephrine, and insulin. So we said before that glycogen, when it's broken down into glucose, the rate-limiting enzyme for this is going to be glycogen phosphorylase, and when glucose is converted back into glycogen via the process of gluconeogenesis, the rate-limiting enzyme for this is going to be glycogen synthase. So glucagon, epinephrine, and insulin affect either the process of glycogen breakdown or glycogen synthesis by affecting the activity of these 2 enzymes. And the way that glucagon, epinephrine, and insulin affect the activity of these 2 enzymes is either by phosphorylation or dephosphorylation, and we're going to discuss this in a bit more detail in the next few slides. So the first thing that we're going to discuss is epinephrine and glucagon. So epinephrine and glucagon are stress hormones. That means that they're going to be released in times that the body is under stress, like fasting or starvation. So during this time, the body's going to want to increase the glucose level in the blood. So it's going to want to break down glycogen into glucose molecules for the body tissue to use. So the net effect of glucagon and epinephrine is that it's going to lead to the phosphorylation of glycogen phosphorylase and glycogen synthase. Now, the thing that is important to understand is that the phosphorylation of these 2 enzymes has different effects on them. So the phosphorylation of glycogen phosphorylase is actually going to lead to an increase in its activity, and the phosphorylation of glycogen synthase is going to lead to a decrease in activity. So when glucagon and epinephrine levels are elevated, they're going to lead to the phosphorylation of these 2 enzymes, and they're going to lead to an increase in activity of glycogen phosphorylase and a decrease in the activity of glycogen synthase. Now, the increased activity of glycogen phosphorylase is going to promote glycogen breakdown. Okay, so now we're going to look at the effects of epinephrine and glucagon on the cellular level. So when epinephrine or glucagon bind to their respective receptor, they're going to stimulate adenyl cyclase. When adenyl cyclase is stimulated, they're going to increase the cyclic AMP, and it's going to lead to activation of the enzyme phosphokinase A. Now, from the name phosphokinase A means that it's going to phosphorylate. So phosphokinase A is going to phosphorylate an enzyme called glycogen phosphokinase A, and it's going to activate it. The enzyme glycogen phosphokinase A is then going to phosphorylate glycogen phosphorylase. Now, remember, glycogen phosphorylase is going to be the main enzyme that is used in glycogen breakdown. Phosphokinase A is also going to phosphorylate glycogen synthase. Now, we said before that When glycogen synthase is phosphorylated, there's going to be a decrease in its activity, and when glycogen phosphorylase is phosphorylated, there's going to be an increase in activity. Now, the increase in activity of glycogen phosphorylase over glycogen synthase is going to promote glycogen breakdown. So now we're going to talk about insulin. So insulin is an anabolic hormone. So that means insulin is going to be released when the body is in a state of high energy. So for example, after a person has eaten a meal, there's going to be high glucose in the blood, and insulin is going to be released. So insulin as an anabolic hormone is going to promote the conversion of that excess glucose into glycogen to be stored for future use. So the way that insulin works is that its net effect is going to lead to the dephosphorylation of the enzyme glycogen phosphorylase and glycogen synthase. Now, of course, this dephosphorylation is going to have a different effect on its activity. So the dephosphorylation of glycogen synthase is going to lead to an increase in the activity of this enzyme, and the dephosphorylation of glycogen phosphorylase is going to lead to a decrease in the activity of this enzyme. So the increased activity of glycogen synthase is going to promote the conversion of glucose into glycogen for storage. So now we're going to look at the insulin effect on glycogen synthesis and breakdown down to the cellular level. So when insulin binds to its respective receptor, remember that insulin is a tyrosine kinase coupled receptor. So it's going to activate tyrosine kinase. Now the tyrosine kinase, of course, is going to come along and it's going to phosphorylate protein phosphatase 1. Now the protein phosphatase 1 is going to go along and remove a phosphate group from glycogen synthase. The protein phosphatase 1 is also going to remove a phosphate group from the intermediate glycogen phosphokinase A, and this, of course, is going to prevent the phosphorylation of glycogen phosphorylase. So the net effect of insulin when it binds to its receptor is going to be the dephosphorylation of these two key enzymes. Now remember that we said that the dephosphorylation of glycogen synthase is going to lead to an increase in activity, and the dephosphorylation of glycogen phosphorylase is going to lead to a decrease in its activity. So the predominant enzymatic activity is going to be glycogen synthase, and glycogen synthase, of course, is going to promote glycogen synthesis. Okay, shifting gears a bit, now we're going to talk about glycogen breakdown in the muscles. So it's important to know that when a person starts exercising, we can't wait for our hormones to increase in order to stimulate glycogen breakdown. So we can't wait on glucagon or epinephrine to increase in order for our muscles to be able to utilize the glycogen inside of them. So this is where the calcium-calmodulin complex comes along. So when muscles start exercising, of course, it's going to release calcium and calmodulin, and calcium and the calmodulin complex is going to activate glycogen phosphokinase A. The glycogen phosphokinase A is then going to phosphorylate glycogen phosphorylase. And remember that we said when glycogen phosphorylase is phosphorylated, it's going to lead to an increase in activity, and that's going to promote glycogen breakdown inside the muscle. So it's going to lead to rapid utilization of glycogen inside of our muscles.